they produce typical structures inside the root. These structures include arbuscules, vesicles, hypha, coils, and outside the cell they produce spores. So these spores are sometimes by some species are produced inside the root also. So but the mainstay or main thing on the basis of which we say this is arbuscular mycorrhiza is the arbuscules. They are the sites from where the nutrients are taken up by the plant, and the soil delivers the nutrient, the fungi delivers the nutrient here. So arbuscule is the main thing, and I just got this picture where it shows, you know, how arbuscule is so active. When arbuscule is present, it shows that this is live mycorrhiza and mycorrhiza is functional. So talking about this mycorrhiza and the glomeromycorrhiza, the scene way back was what? We used to identify them on the basis of morphological characters, meaning I remember in my lab I used to sit, isolate them from the soil, culture them, and then under the microscope I used to look for spore color, make the proper preparations, surface texture, subtending hypha, wall layers, and then I used to give them a name, right? And we used to identify several species. We have a lot of species reported from the world, like in India, we have reported 167 species by now, but they were classified on the basis of morphological characters mostly. And I tell you, according to my study, where I surveyed all the studies done in India, there's hardly, very few people are using molecular methods even now for classifying or identifying their fungi. So these are some examples that they look beautiful, these spores of these fungi, but they are difficult to identify, meaning very often in the literature we observe that the identification is wrong. And again, the other fungi I could identify by culturing, but it's difficult to culture them, meaning you can culture them, of course, by root organ culture or in the pots, but that's a lengthy and tedious process, meaning when you are going to study 160, 150 species occurring, it's hard to culture them, cultivate them, and study them that way. And say the other problem which we felt is that the single spores, the single species, it produces spores of different colors, different morphology in different circumstances. That leads to wrong identification. So there were actually very limited features on the basis of which we could identify them under the microscope. And then the spores, again in the environmental problem, when I was studying mycorrhizosphere, I realized the spores which I isolate from the soils is way different from what it is present in the culture, because in the soil it's interacting with a lot of other microbes, and its shape and face is absolutely different than what we see in cultures. So we have other problem is that we have very few experts in the world which who can actually identify them and confirm that. Usually in the papers you would see wrong classification. So then because I was working on mycorrhizosphere, and I could not identify them. I just had to do the quantitative study. But what I observed is that there's a positive correlation. Whatever AMF is inducing in the rhizosphere is mediated through root exudation, and it correlated with the other fungi present in the mycorrhizosphere. This paper of mine is quite well accepted, meaning I have even now 124 citations of this paper. So finishing this one till here, I was doing most of the studies based on the culture, and I used to culture them and observe them under the microscope and try to identify them. And we were confused because the classification was not accurate. And that, I would say, was a season, because I'm just trying to convince you that it was really a beautiful journey, crossing the different fields, different disciplines, and reaching to where we are now. So with this one season of my studies finished, and I diverted the season 2 where I talked a lot about molecular biology methods. I didn't do much. I tell you what happened at this stage is that I began with sequencing single specimen, meaning we used to culture in our lab these spores, and ideally the success rate for this culture is just 2%. And you cannot do DNA isolation from the field spores. I told you already why, because they are already infected with other organisms. The spores are huge in size, and when you isolate them from the field, you would see that it is already infected by the other organisms, and that is going to lead to the wrong result. So we used to culture them, and we used to then isolate DNA from there, and then amplify, sequence, and then we used to identify and confirm our identification. Then in the evolution of next generation sequencing, which I'm going to focus and take you further, we had several steps, like we had parallel Sanger sequencing, we had second generation sequencing, we had third generation sequencing platform. The second generation gave us short rates, the third generation gave us long rates, but they were prone to errors. So with all these methods, being in a college, and I was quite interested, but I always lacked facilities. I could manage sequencing and isolating DNA from the spores in the lab, but again, the beauty of the journey came. God came to our rescue, and there were lots of companies at that time who were doing this work. They just wanted money. You isolate DNA from the sample and give it to them. They would do all sequencing work for you and give the sequences. So I didn't have to bother much after that, and I was given sequences, and this way we could get up to 237 sequences belonging to many species. So we didn't use the ITS primer because that doesn't give you right result in case of our fungi. We used SSU, ITS, LSU primers for amplifying these sequences, and this is a multiple sequence alignment which I made for a huge number of species, and this shows you the conserved region and the variable region in these sequences. But when I was working with so many sequences, it gave me special interest. And when I made a phylogenetic tree with these sequences, this is again a phylogenetic tree which I have drawn with the software in the ITOL. I tell you, by this time I had developed a great fascination for softwares, applying information technology tools in these studies, and overcoming my limitations of the facilities in the laboratory. And apart from facilities, what happens is, doing a genome sequencing project is required huge amount of funding and money, and really you cannot play with that. You cannot afford to spend that much of money for unless it is actually needed. So I started enjoying this, and I drew, but there were lacunas in this. I realized there are problems, and it's not up to the expectation. So after this one, what happened, that I still thought I need to learn how this tree is being made, and there's some problem with this one. I said I already developed a huge interest in information technology resources by now. I liked the softwares a lot. So I tried searching for softwares. I shifted to computers. I did a course on networking, hardware, software, lots of languages. And then I gradually tried to understand what is happening with this in this computer program and how these things are happening. So let's not divert to computer. I am again back to my sequences. And what happened with this high throughput sequencing technologies? We had flood of sequences. Like, we really could not afford anything without computational programs, and meaning understanding where is the error, what is the quality of this sequence. If your sequence is wrong, you are going to lead to wrong results. So with this, that was the season I explained to you already with computers. And with this one, I had trained myself to work with a lot number of sequences. I was very much friendly with computers, softwares, algorithms, meaning at least I could understand and try to apply that in my studies. So shifting to microbiomes, and the first thing, I just like to define this for the students who are undergraduates here, is this was in 1998, Jo Hanselman, she first used this term metagenome. And she said, like genome of an organism, we could say metagenome from all the sequences isolated from one environment. So she was the one, but what do we understand by metagenome? Metagenome refers to all the sequences which are isolated from one environment. This is how it was defined. And by now I had understood that I am going to shift now from mycorrhizosphere to rhizosphere microbiome. And again, it was a beautiful journey. So Lord has trained me with lots of softwares by now, lots of sequence experience, and the problems which we were actually facing when we were working with our fungi. So that was my next paper. And my rhizosphere was now what? My rhizosphere now was a root with exudates around, and it has 14,000 genes of protozoa, it had 14,000 genes of arthropod, it had 13,000 genes of algae, it had 6,500 genes of soil bacteria, it had nematodes 18,000, and my fungi were just a part of 15,500 fungi which I could decipher, and this included all arbuscular, ectomycorrhizal fungi, pathogenic fungi, and everything. So this image of rhizosphere I had in this season, and but I was still till here. I always thought that before we begin with talking about this rhizosphere microbiome, these three terms, meaning of these three terms should be absolutely clear to everyone. Like, what is metagenome? What is the difference between metagenome and microbiome? What is microbiota? So by definition, microbiome refers to the microorganisms and their genes together. And microbiome also includes the theater of activity which is happening in that region. It's a dynamic thing, and it is an interactive micro environment, meaning micro ecosystem that can change with time and scale. So microbiome is the dynamic entity which refers to the all the organisms present and the environment and whatever is happening in that environment. But on the other hand, metagenome just refers to the genes and sequences. Microbiota, which we used to study, it refers only to the microorganisms or microbiome, right? So these are the three things which sometimes, I feel that they appear to be confusing. So when we reach to metagenome, we could talk of any metagenome. The concept was same everywhere. If you talk of, for metagenome, the most popular work or the favorite work which has been studied in a lot of details is the gut microbiome. And you relate this with your gut feeling, it is related to the microorganisms present in your gut. So we went to the environmental samples, and it could be any environment. For us, it was rhizosphere, and in the rhizosphere microbiome. Now I would talk about what? How much in this pie chart? How much is that bacteria? How much is that fungi? How much is arthropods? And that was it. And I could do it at the different regions on this soil. So after having understood all this, still, we had a lot to go, and there are certain concepts I thought before I show you actually how we analyze these data and infer meanings out of this. I thought it makes sense to explain a few more terms here. One is core community. Like I tell you, first thing here again, talking about the terms. First term we must understand by now is holobiome. What is holobiome or the superorganism? The scientists by this stage, by this season, started saying that we should not talk about the plant alone. If you read in the newspapers, you would say that they say that we should not talk about a person alone. It is associated with a lot of organisms, so we should include that also in every treatment. This is what is actually happening in the medicine now. And same for our rhizosphere. So we will believe that it's a superorganisms, and it is said to be a entity for evolution, meaning we should always take the plant along with the associated microorganisms, not only root, but the leaf and the other parts also. So I was telling you about this holobiome or the rhizosphere microbiome. What happens is that it has millions of the organisms present. And when we started studying them, we came to know that we can classify them into several types of communities. We could meaning divide them. One is a core community, meaning if you isolate rhizosphere from 10 plants, you will see them present. In all healthy 10 plants, this core community is present. Then what you could decide is that they are very important for the plant to exist, for a healthy plant. Then there was a minimal community, like species 1 and 2 in this case. They are always present, and if you remove them from there, the plant is going to die. So they are necessary for the existence of the plant. There is another type of community in the microbiome is accessory community. What is that? That refers to the, I have drawn this with 5 to 8. All these figures I have taken from that paper. So 5 to 8, what does that mean? This accessory microbiome, 5 to 8 means they can be easily replaced by beneficial microbiome, and 9 to 11 is synthetic microbiome. So beneficial microbiome. So what did it teach me and what scope does it give me was that once I understand what all is present in the root microbiome, I can easily play, I can play with accessory microbiome. I cannot touch the minimal community, the organisms, the microbes which fall in the minimal community. I cannot touch the core community, but I can play with accessory microbiome organisms, and I can replace them with other beneficial organisms, maybe any beneficial organism. And with that, the term came synthetic microbiome, that we can synthesize microbiome. This gave birth to a new field that is called as rhizosphere engineering. Now, what is rhizosphere engineering? See, I tell you, these are the seasons, and why I found it beautiful that from basic biology, we were botanists, when we were studying, we were students, we didn't have any distinction into molecular biology. We didn't have any distinction in, we didn't bother about computers. There were no computers at that time. And we had no distinctions. But now we talk about rhizosphere, and engineering has come in between, and molecular biology, bioinformatics tools are so advanced. This happened during the journey. So the rhizosphere engineering became a full-fledged discipline. And we could now, when we analyze a microbiome in the rhizosphere or in the gut, we could say these are the good friends, Facebook friends, these are good friends, these are bad friends, these are ugly, they are going to do anything wrong, they will not do anything God. Like, we can manage bad friends, but we cannot, we need to be scared of these ugly people. So that was this rhizosphere engineering. And I said already the same concepts were being applied to the human microbiome. Basically, all these concepts are coming from medicine, human microbiome, but the environment is as important as human, so they are being applied here also. So in this season, what I learned is it's a huge branch which is coming up, and you can play with lots of things. You can fiddle with the microorganisms, and you can synthesize many things with these sequences, or you can at least plan and model things in this era. So I reached to the next season with this one. In my journey was the metagenomic analysis. I thought, unless I understand how do you analyze this, how would I know which community is important? And my question to myself was, can I apply this in my mycorrhizal fungi? Can I apply this in my mycorrhizal fungi? So the next season began, and I was determined to know in my rhizosphere by metagenomic analysis who all is present, meaning with my fungi, who all organisms are present, what are they doing there, meaning how they are interacting with each other, meaning what is the cycle in the interaction. And I was trying to, tell that how they are interacting, what enzymes are involved, and what is the structure of these enzymes. So I was trying to answer these 3 questions by metagenome analysis in the rhizosphere microflora. And by now, in my journey, everybody knows this, that I'll talk only about the sequences. Microorganisms, which we observe under the microscope, have been left way behind, right? And so I will call a species what? The species in molecular biology, in genomics, is OTU, Operational Taxonomic Unit, meaning they believe one sequence refers to one species. So one sequence is OTU. We must know OTU, and I used to be concerned only with the OTUs, like which all OTUs are present and which species this OTU refers to. And the environment which I was studying, I said already, was rhizosphere. So I will, it might become a bit difficult, but it is quite interesting. I have tried to simplify this. And let's see how it goes. But I'll be a bit quicker because I cannot afford to take details. But I just wanted to show the students that how easy it is. Though the concepts, once you learn the concepts, doing things practically is not that difficult. So these are the steps which we take in doing the metagenomic analysis of any community in the rhizosphere. So how do we study this? We'll prepare the sample and we'll do DNA isolation from the sample. We'll do the DNA amplification. We'll do the quality check. What sequences we have got, are they okay, or there are some wrong sequences giving to the wrong results? OTU picking, that we should know how many OTUs are present here. And then we'll taxonomically assign those OTUs, meaning which species this OTU refers to. So broadly speaking, these are the 5 steps. And I will just quickly take you through these 5 steps. There are lots of softwares in the market for this one. If you see at the bottom, my cursor, yeah, it might work. Yes, it is working. So you can see there are lots of softwares, but I thought for the students who are born in the computers, the first year students, second year students, they have come to the world when computer is everywhere. They cannot live without computer, and there are a lot more things in the computer. I picked up the software which is very simple, and I thought after this lecture maybe students would like to fiddle with this. That can we work with the theoretical data and try OTU picking in that. So I picked up the simplest software and the most popular one, that is QIIME. So we'll use QIIME in this next few slides and we'll see how we do it. But I said I'll be a bit quicker because we might miss some part of the journey, but I tell you it was very interesting. So starting with the sample preparation. Sample preparation is easy. I said I work in a college, so at every step, God was connecting the dots and was easing things for me. So sample preparation, the soil isolation, DNA kits are available. I had no problems. And we realized we need to run 3 types of controls here. One is negative control, where the soil sample is absent. One is positive control, where already known existing microflora is there in the sample, so that we can match. And another type of control which we want to run in this, because there are a huge number of sequences and it is prone to errors, is another type of control which we run here is mock community, meaning we have synthesized microbiome, I told you already. We have synthesized community, which is standardized and supplied by several companies now. And then you can know that you already have predefined microflora occurring there. So that was the first step. I said it was made easy by the existing situation. Then the next was deciding for the primers for our fungi. The primers, usually for fungi, we use ITS primers, but these ITS primers don't work good with AMF. Why? Because we cannot resolve the organisms up to the species level, only up to genera if we use ITS. But if we use SSU, LSU, and ITS primers together, the fragments of those, it works. And we could, as I showed you in my previous pictures, we could resolve them to the species level when we use these 3 primers. So that was about DNA amplification. And there's one more thing about DNA amplification. I'll tell you in the next one. So what happens is after DNA amplification, what we get, meaning let's come to the practical terms. The DNA amplification is done, and what we get out of that, we get 4 files in my this thing with which I am going to work. One is a mapping file, the other one is barcode file, third is sequence file, fourth is the reference data file. I'll quickly tell you what these files contain. The mapping file is the file which has the roadmap, meaning it tells you all your hypotheses. What is your experiment? It has several columns, right? Can you see these columns? I hope you can see these columns. And what is happening is that in this example I've taken this from a human microbiome, where 2 individuals are being studied. we have four body parts which are studied, and we have 5 time points in here. And this is the mapping file, a typical mapping file which has all information present. Then we have another barcode files. This barcode file is also very important, and in this mapping file also they give barcode sequences. Then third, we get the sequence file. Sequence file is the sequences which you have collected after amplification. And we have another file that we call as reference data file. What is this reference data file? Is the already identified sequences. Like say for example, in my case, I plan to identify AM fungi. So I will not use the reference data for bacteria, right? So reference data file, we have to decide which file we are going to use here for identification. So after amplification, I got 4 types of files, and I'm going to start with these 4 types of files. I said I'm working with PAN. Because we have millions of sequences in this sample, and we got many sequences, and there will be a lot of confusion, a lot of things happening with these genomic friends. And so the quality control is the most important thing here in metagenomic sequence analysis when you are doing. And here comes the term barcodes. So barcodes are what? Are the special unique identifiers which we add to primers, so that we can identify our sequences to which where they are belonging to. And these barcodes and these primary sequences, they need to be checked, meaning they should not be present in the sequences which we are using for taxonomic assignment. So these are the quality control. I said it is quite important. And yeah, I had written this for you. It says quality control and contamination check is a must, and you have to be expert in that. So what you do in quality control check is, I have just brought a small picture of the software with which I work, CHIME, and a bit difficult for our generation when we are biologists to go to this sort of working, but for young students, I'm sure it will not be difficult. So CHIME is not GUI. It is not GUI, it is a command-based system, but this quality check, CHIME can do it for you. How it does it? It does the quality. It has programs inbuilt programs which will do quality filtering, and the quality. You have to check by your eyes also sometimes at your sequences wherever you feel doubt that is there a wrong base has come, or is there a gap or anything. But we have lots of softwares with that. You can do this quality check of your sequences. Then we have to do the multiplexing, meaning we have to remove the barcode and the primer sequences which have come in the sequences which we are going to use for taxonomic assignment. So this quality check is a very critical step, and you can always practice for this in your laptops. You have lots of dummy data, mock data present on internet. You can pick up any data, install CHIME in your laptop, and start playing. So let me show you how do you do that. That's my computer, and what I did is, let me share my computer. Yeah. So I have this Ubuntu installed in my computer. Yeah. Ubuntu, is Windows friendly DOS prompt, right? So what it has fixed commands. If you go for this, it has fixed commands. Sorry, it is repeating itself. I'll just show you 2 steps that how did I do it, and then maybe you can practice on your own. So CHIME is already installed in this 2021.4 version, and I passed this command already written in my laptop to activate this. Now CHIME is functional in my laptop, and I can activate this. And I had fixed commands. You see this command, when I pass, it will demux my sample, my sequences which I had got. So I hope it's not getting too much, but I find it very interesting and beautiful. So I'll carry on with this further. And if you want to ask anything, you can ask after this one. So the next step is sequence clustering and OTU picking, meaning whatever millions of sequences we had, we'll cluster them with an algorithm and we will get something like this. So you had many sequences, it will tell you this sequence 8,200 times and 16 samples. So this gives you all quantitative data and develops a feature table, meaning it divides your sequence, categorize your sequence based on the features. And what do you do after this? You have got this category, you run your reference data, and on the basis of similarity to that reference data, you say that these were these bacteria, these fungi, and this is how you get a picture. And then you can do any statistics, any graph with these information which you have got. So these are already inbuilt databases in Chime. You can select whichever you want. But for my yeah. So with this one again, another season finished last year when we were absolutely online. And last year, I remember just before the lockdown, 1 year before the last season and the present season, which is the season of reference databases, I was just worried and concerned about the reference database, which I was using for studying my AM fungi, and I just realized the database is usually used by Chime and the other softwares. I mentioned some names here, but we use Marza AM for AM fungi. So what were the problems when I used this database for identification is that it limits the sequence on the basis of SSU sequence collection, doesn't talk about ITS sequences, and ITS sequences remain very purely curated for AM fungi. And what was happening, I told you the reason already, the sequences isolated from the environmental sample showed very poor matching. And we require, we couldn't match many of these sequences, a large proportion of the sequences remain unidentified, and still it required lots of specialized knowledge by very few experts. We had the cases of new species occurring, those were not present in the databases, or rare species, we couldn't identify them with these databases. And we realized that we have a need to generate a training data. And the quality of data was another limitation. The type of data present was not of good quality. We couldn't trust on that. So yes, there was one more season which has come recently, is artificial intelligence. So these kids, they know a lot more about artificial intelligence. What is it doing? It is everywhere. So if you talk about the future, but I tell you how artificial intelligence came in my work was, I realized is that I was just studying artificial intelligence because I keep understanding lots of things around. I realized that this species identification with these metagenomic databases, metagenomic data analysis method has reached to quite an acceptable level. I was just searching, I read lots of papers, analyzed several papers, and I realized that it is getting automatized with artificial intelligence methods. So what I'm trying to tell you that in this paper of mine, what did I observe? That these steps in identification of taxa in metagenomic data, like your Google search is getting automatized by several artificial intelligence methods being applied at every step of metagenomic data analysis. We can't do it with AM fungi, we are way behind. But with this knowledge, what I realized is that communication among the scientists of different disciplines is the key to solve the present day problems. And we must encourage our children, our students to be multidisciplinary rather than focusing on one discipline and being only the maybe mycologists, because once you have a better understanding, then you can think of more solutions to a problem. So that was just a picture. I've taken this from this paper in 2012, and this red machine learning algorithms I have added because I realized a new era, a new season is going to come where machine learning or artificial intelligence is going to automatize all the steps in this analysis. So with that, I come to an end of this beautiful story, beautiful journey of mine. And I say this with proud that this was one of the most beautiful journey in my life. And embracing microbes along with computers was much like joining the dots of Steve Jobs. So with this, I would like to thank you for your patient listening, and I would be very happy if anybody has a query or anybody wants to uh,
Thank you
know.
so much, Manju. Thank you really for showing us how actually multidisciplinary techniques work so much better than just sticking to your own line of research. So I hope the children, the kids take a message home, and that they do want to, play with science in a way, play with the whatever techniques are available these days, and get ahead. Any questions?
Pleasure was mine. Thank you, Anupma, for providing me a
Not at
platform
all.
to talk to the students. And your college, actually, I see is multidisciplinary. You have biomedical sciences, you
Yeah,
have
yeah.
computers, you have chemistry, you have lots of things. Like I
Yes.
don't get these sort of students in my college, so that is why how I thought that this is really going to benefit your students. Meaning how I would encourage them to make friends with computers, make friends with electronics, because if you have a problem, you can talk to them.
Yeah,
Our knowledge is
definitely.
not in books. Friends also have lots of knowledge. Yes.
Yeah, yeah. Actually, we have a summer internship program called Elite, in which we encourage only interdisciplinary
Oh,
projects
that's very good. Yes.
for, yeah, for the 2 months of the summer vacations. And we have at least three teachers from different departments who float projects, and interested students apply, and then they are given fellowships for the 2 months.
Okay.
But
So
yes, since this
would
corona
you give this
lockdown,
to our students also? Meaning anyone
uh,
can apply, or it is for your students only?
it is for our
Our
students only,
students.
actually.
Okay.
Yeah, but these past 2 years it has gone waste only
Yeah.
because of
Yeah, I
corona
think so. We have
complications.
just crossed the institute level, crossed the countries.
Yeah,
I'm very much in touch with the all has come because of this technology now. Yeah.
yes, definitely. It was really a mind-opening kind of a talk.
Thank
Shruti,
you for having me
thank you.
here. Yeah.
It's our pleasure. Um, there was a question, I think, Shikha Srivastav, she says, when we say gut microbiome, is it right?
Yeah, absolutely
Yeah.
right. You can say fecal microbiome, you can say intestinal it has to be one environment. That's it.
Mm-hmm.
For
Uh,
microbiome, the constant feature is the environment should be same.
right, right. Shruti, can you take over asking the questions, please?
Yes, ma'am, sure. Ma'am, there's another question asking what are precautions do you suggest for isolation of spores from roots in order to get quality?
From the rhizosphere. In order to get quality, I tell you,
Yes, ma'am.
when you are isolating spores from the rhizosphere, you should meaning you should observe to for quality. Yeah. So what you should do is you should observe them thoroughly. You will find that the spores which are old or which are infected by bacteria, they will be dark in color. They will show you dots over them. So you don't have to pick those spores. You pick only the spores which are live, vibrant in color, and they show you live cytoplasm. So while picking, you will always realize that you can distinguish between the two type of spores. The one is dead, which is having lots of dots in black color. They are dying, and some are vibrant with live cytoplasm. So you got to pick only those spores which are live. That is the main precaution. Otherwise, you will not be able to identify, do nothing with that.
Okay, ma'am. There's another question also.
Yeah.
On what basis we can differentiate beneficial microbiomes and non-beneficial microbiome on basis of their genetic sequence?
See, we cannot distinguish this on the basis of sequence. See, I told you we are trying to answer three questions. Who all is present? It's just like your class. See, you have a class of 50 students, and you are observing all class. In microbiome, you are observing all these 50 students, right? And you would see that Two students, if they are removed from the class, they will not be able to, this class is not going to work, right? And few, they are quite important, but few, if you are going to take this class to the best level, meaning synthetic, you said, if you replace this was not the natural setting, but if you replace these two microorganisms with the other ones, you can do that. You insert those. Can I tell you, I'm working with some scientists in IIT, what they do? They have some bacteria, beneficial bacteria, right? And inserting a rhizosphere, in the rhizosphere, inserting a beneficial microbiome means what? You take some bacteria and insert them in the rhizosphere. So they will change. If a new student comes to your class, it is going to change everything, isn't it? So it is just an interplay, same sort of interplay which happens in your class. Some students are must, some students are in a normal course. There is everyone there. But if you are going to take it to best, what will you do? You will synthesize. Some students are sick. They are going to spoil this class. That you will remove them. So we cannot say a microbiome is sick. If meaning you can say when you have a lot of sick students, you will say that class is sick. But it just all depends how function. Meaning it all is determined by the function of the sequences present. These sequences are expressed. They're always expressed, isn't it? Yeah, you agree with me? Sequences are expressed. So with the protein they are synthesizing, you can know what function they are performing, and you can always know how they are interacting, where we can change. So you can always predict their function. A good function is coming out or a bad function is coming out. I showed you that picture.
Okay, ma'am.
Yeah.
The next question is, do we hybridize a mycorrhizae? And if hybridized, what chance does it induce on plant in which it grow?
So what do you mean by
Can we?
hybridizing? What do you mean by hybridizing mycorrhiza? Can you just clarify, please?
This question is by Aishwarya.
Yeah,
Aishwarya,
Ashwarya can.
bolo.
What do you mean by hybridizing? I don't understand. We do a lot of hybridizing work with plants, and mycorrhiza also comes into the picture, that we study that how this hybridization is changing the mycorrhiza in the rhizosphere, the beneficial organisms in the rhizosphere. But we don't try to hybridize mycorrhiza. Yeah.
Ma'am,
Yeah.
another question is we have, can we use rRNA sequencing for the better classification of the different fungi? And if so, can we say it will take less amount of time that sequencing the whole genome of the particular fungi?
So you said rRNA, no?
Yeah.
Yes, ma'am.
Yeah, so the ITS I was showing, I was not talking of whole genome sequencing. I just showed you one sequence, and that all the study which I showed you was by the rRNA sequences only. I didn't do whole genome. I just took one barcode sequence, that was rRNA sequences. Yeah.
Okay, ma'am.
Yeah, that is certainly going to happen. This whole genome sequences, how expensive it is. Nobody will give you money to do that. We have to find conservative ways, and that is why we do rRNA sequencing mostly. In most cases,
Okay, ma'am.
especially for our work, it's sufficient.
Yeah, we have to devise of methods in which we can financially also, be
Yes,
able to do it. Constraints are there of all types,
yes, yeah,
techniques and finances and availabilities and all. Okay. I'll be talking to you later also, Manju, about my own
sure.
kind of
My pleasure. Yeah, yeah, my
right.
pleasure.
Shruti, can you
Yes,
wind up
ma'am. Yes, ma'am. Sure. Thank you, ma'am, for your enlightening speech. I would like to thank all the staff members and the lovely audience for making this event a grand success. Once again, I thank one and all present here. Thank you.
Yeah,
Thank you.
thank you everyone for having
Thank
me
you.
here, and I enjoyed talking to you guys. Thank you very
Thank you,
much.
Manju.
Thank you, Anita. Thank you. Yeah.
Okay,
Do we
thank
leave now?
you.
Yeah. Thank you,
Yeah, you
Nopma.
can.
Thank you. It was lovely
Okay, bye
seeing
bye.
you. At least online we
Yeah,
could
I
meet.
know, I know.
Yeah.
Yeah.
Thank you very much. Okay.
Okay, welcome. Our pleasure. Bye.
Yeah. Thank you.
So, chalo, then we can end the meet now.
Hmm.
Yeah.
Yeah.
Okay. Thank you everyone for attending. Thank you, Janki ma'am.
I think she's left.
Thank you, Shantanu. Thanks, Vijay. Okay,
Namaskar, ma'am.
then
It
see
was
you.
a lovely talk.
Huh.
Nice and very informative.
Thanks, Shantanu.
Thank you,
I
Shantanu.
talked to her. Na, she has combined so many fields, so I thought we should give the students a chance to know that you can actually be interdisciplinary and it will work.
Absolutely, ma'am.
Yeah, yeah. Okay, thank you for being there. Anita, should we end the talk now?
I think
Ah,
people are
yeah.
leaving already.